So our last journal club was about medical assistance and dying. And that was a really important topic. And as we know, no surprise, the major demographic predictor for accessing that service is advanced age. And that was followed by grand rounds at Maine General, which focused on cognitive impairment and dementia. And again, of course, no surprise, the main demographic predictor was advanced age. So I'd like to continue that theme in today's talk, but from a different perspective. Instead of looking downstream at the medical and cognitive consequences of advanced age, I'd like to look upstream at the biological causes of aging and to see if we can find out there some clues for how to live a long and healthy life, and perhaps set up a research program for expanding on those clues. But I don't just want to review current theories of aging, because I think if they were all that good, we'd be living longer than we do. So I'd like to propose that we take a new approach. See, here's the thing. In every cell in the body, there are 6 trillion chemical reactions per second. That's why it's so hard to make progress. The complexity is unimaginable. But it turns out there are only 15 to 20 chemical reactions that comprise what we could call core chemistry, and it's the same 15 to 20 chemical reactions in every cell in the body. So if we approach aging and ask how does it arise from core chemistry, and what can we do about those changes in core chemistry that bring about aging, then our project is much simpler, much easier to deal with 15 to 20 chemical reactions than 6 trillion. But there's another reason for approaching it this way, and that is that it's very likely that the chemical reactions of core chemistry govern everything else that goes on in the cell. So any progress we make at the level of core chemistry will propagate to the rest of the cell and throughout the body. So if we want to get things done, this is the place to look. Now, before I describe this project, I'd like to describe some of the background for me. And it goes back to the early 1960s. I remember the early 1960s. And I do. And I remember that back then, if you were male, old age began somewhere between the ages of 45 and 52 years. I knew this because that's about the age when you had a heart attack. And if you survived that heart attack, then sometime in your 50s, you had another heart attack. And if you survived that heart attack, if you just had a strong constitution, then sometime between the ages of 60 and 68, the farthest reaches of human lifespan, you had your third and final heart attack. And that was that. In the 1970s, I was taking a nutrition class, and you could feel the intensity of research trying to understand why heart attacks were so epidemic. A lot of suspicion then fell on a diet high in saturated fat and cholesterol, but everybody knew that a lot of pieces to the puzzle hadn't been discovered yet. I was also taking a food science class. Now, you might think that a food science department would have a lot of ties to industry, and you'd be exactly right. And on one particular day, we had a guest lecture from a representative from the dairy industry. And he wanted us to know that all of that talk about avoiding dietary saturated fat and cholesterol was just a bunch of nonsense. That you could have all the heavy cream and butter and whole milk and eggs that you wanted. It wouldn't do you any harm, and you'd be supporting the dairy industry. Now, his views were not widely accepted then. In fact, he was the only person I'd ever met who actually seemed to believe that. But he made 2 points to back it up. First, he pointed out that all of the data implicating dietary saturated fat and cholesterol with heart attacks in people was correlational. If you really believed in the hypothesis, you'd do a proper controlled trial. Half the people would be randomized to consume a diet that was supposedly bad for them, and you'd follow them carefully over time to see if they died. The professor was at the front of the room, standing off to one side. He was getting kind of uncomfortable. He couldn't tell whether he wanted to interrupt the talk at this point. Apparently it had not been vetted first. But the speaker went on. He said, you could do that study, but it wouldn't show anything. It was impossible to prevent heart attacks, and it was foolish to try, because heart attacks were not actually a disease. Now, polio, that was a disease. It was a terrible disease, but we knew how to prevent it. And tuberculosis, that was also a disease, a horrible disease, but we knew how to treat it. And even though not every disease could be prevented or treated, this was 1978, it was just a matter of time. And when all diseases were cured or prevented, everyone would be able to live out their natural human lifespan of somewhere between 45 and 68 years. See, heart attacks back then were universal. The only way not to die of a heart attack was to die of something else in the meantime. And so heart attacks, the speaker said, were just the natural end to life. So if you were 45 and you died of a heart attack, that was a privilege. That was a sign of the progress of civilization, because it meant you survived all diseases and you were able to live out your full human lifespan of, in your case, 45 years. It was a remarkable talk. In order to support the dairy industry, he was actually coming out in favor of heart attacks. It was amazing. Of course, as we know, the medical research establishment did not take their direction from the dairy lobby. And over the last 45 years, there's been tremendous progress against what we very comfortably now refer to as heart disease. And those words of the dairy lobby all those years ago are now entirely forgettable. Except for one thing. Everything that he said about heart attacks then, we say about aging now. And so, if you're 96 and you can't see and you can't hear and you can't walk and you can't remember, that's a privilege. That's a sign of the progress of civilization, because it means you've avoided all the diseases, allowing you to live out your natural human lifespan of, in your case, 96 years. But why exactly are we correct when we say that about aging when the dairy lobby was wrong when they said it about heart attacks? What makes aging different? Is aging qualitatively different, say, the soul gradually leaving the body? Maybe. But if the soul is gradually leaving the body, it's surely because the body is becoming biochemically less hospitable to it. It must trace to biochemistry. Or is it because aging is vastly more complex than heart attacks? Maybe. But our tools are more sophisticated than they were in 1978 also. But that belief that aging is more complex, we don't actually know. It certainly looks more complex on the surface, but that doesn't mean that it really is. There's a line of evidence, fairly strong as far as it goes, that 70% of the variance in aging can be causally explained by the progressive malfunctioning of a single gene. The complexity of aging may turn out to be illusory. If it turns out that we could do something about aging, should we? My cousin asked me, what are you working on these days? And I said, aging. And she immediately said, I don't think people should live forever. She said, and I'm talking about myself. I certainly have a list of other people I feel should not live forever. And I agree that immortality raises certain medical ethics problems. But what about a more modest increase in lifespan, say 20 or 50 years? It turns out that the demographic implications of that in terms of sustainability are probably fairly minor compared with the rapidly decreasing birth rate around the world. As recently as 1960 in the world, the average family size was 5 children. As of 10 years ago, that was down to 2.4 children per family globally, and it was rapidly decreasing. In the United States, it's 1.7 to 1.8 children per family, so we're below replacement rate. Conversely, are there reasons for doing something about aging? The standard rationale that's given in just about every article on this topic is that aging is the primary risk factor for every major non-communicable disease. And as we saw with COVID, It is the major severity factor for every communicable disease, or just about every communicable disease. And so any progress that you make with aging immediately translates to progress on just about every disease on the planet. So from a research efficiency standpoint, it makes a lot of sense to research aging. But there's another reason that I've never seen written and I've never heard spoken, and to me it makes more sense. To me it's really the bottom line. If we decide that we're going to research each disease piecemeal and stop there, and not try to research aging, then we are saying, explicitly or not, that our goal is to enable a large number of people to reach an old age defined by disability, impairment, and low physical quality of life. Not because we couldn't do anything about aging, but because we chose not to even try. And that cannot possibly be the right answer. But if aging might be tractable, and if it's worth taking on, where do we start? Well, a logical place to start is with what we know kind of works already. Aerobic exercise, endurance exercise, resistance exercise, weightlifting, caloric restriction. It's good to be at a healthy weight if you want to live a long time, and intermittent fasting. And what these 4 interventions have in common is that they're all metabolic interventions. They change the way the body generates and utilizes energy. As I looked into this, I discovered that other healthy things like not smoking or eating a lot of vegetables also exert a lot of their health effects because they're metabolic interventions. They change how the body generates and utilizes energy. So metabolism seems to be pretty important to aging. The second thing that these interventions have in common is that they work pretty well. Which is to say that if you're 40 years old and you run 5 miles a day and you lift weights and you're a healthy weight and you fast 1 day a week and you don't smoke and eat vegetables and the next year you die of a heart attack, we'd say you got really unlucky. That shouldn't have happened. In nursing homes, a healthy diet alone will reduce all-cause mortality by between 85 to 90%. The effect size is very large. The third thing that these interventions have in common is that eventually they stop working. Which is to say that if you're doing everything right and you're 90, you're probably not going to get as much out of it as if you were 40. So from our perspective, that last observation is actually the most useful. For one thing, it provides a way of framing the problem. Instead of just kind of globally saying, well, we'd like to slow the rate of aging, we can ask a much more specific question. Can we maintain the benefits of these metabolic interventions so that they are as effective at 90 as they were at 40? Because if we could achieve that, that would surely produce a significant increase in human lifespan. Looking at it from this perspective also simplifies our problem, because for a behavioral change, say adopting an exercise program, to bring about a physiological change, say a slower rate of aging, there has to be a signaling cascade, a series of chemical reactions that are set in motion by the behavior and that culminate in the desired physiological outcome. And these signaling cascades usually involve only 7 or 8 steps, meaning we just need to identify 7 or 8 enzymes. And if an intervention, stops working, then 1 or more of those 7 or 8 steps, 1 or more of those enzymes, must be downregulated. And there are only about a half dozen ways that an enzyme can be downregulated. And so our project actually now looks pretty simple. If we want to extend human lifespan, we just have to find out what the 8 enzymes are and examine each one for 6 different ways of it being downregulated. Very, very kind of simple way of going about the problem. And so about 18 months ago, I dived into the literature, very optimistic. What I discovered is that not only did we not know why the metabolic intervention stopped working, we did not even know why they worked in the first place. And that was because the entire field looked like a giant rummage sale. There were some interesting findings, but no coherence to it, nothing that tied things together. I'll give you an example. In 2007, a group of researchers at Case Western Reserve University developed a genetically modified mouse in which a single gene affecting a single enzyme for metabolism was altered. It was upregulated. Now remember before I said a single gene may account for 70% of the variance in aging? This is not that gene. This gene, as far as we know, has nothing to do with aging. Notwithstanding that fact, the mice lived twice as long. The reason they lived twice as long is they aged half as quickly. We know this because when the female mice were the human equivalent of 86 years old, they were giving birth to normal litters. This is a huge breakthrough. First of all, it's a big deal to double the lifespan of a mouse. Secondly, genetically speaking, mice are us. We have that same enzyme in our bodies, and in metabolism, it does the same thing for us as it does for mice. You would think this would have been presented as a major breakthrough. It was not. It was presented as an oddity, as a curiosity, as if it belonged in the strange but true section of the news. And here's what's weird. In 2022, 15 years later, to the extent that it was cited at all, it was still being described as an oddity, as a curiosity. In 15 years, there had been zero progress in understanding this breakthrough, or from what I could tell, even trying to understand it. I came to feel that the problem with this field is that a lot of the biochemistry is being done by biochemists. And biochemists hate theory. Theory is wrong. Theory is incomplete. Theory is outdated by the time you even publish it. Theory is what you do when you've lost all creativity in the lab and there's nothing left for you but to sit behind your desk and pontificate. Theory is not a respectable activity of a good scientist. But of course, as mental health professionals, we know that theory is something more than that. Theory is a map. Theory ties together what you already know into a coherent whole so that you can move beyond it. The fact that theories are falsified is not a failure of theory, it's the purpose of theory. And this field lacked all theory. It had no map. As you can imagine, what I'd like to do today is present what I believe is the map, explaining how metabolism governs aging. And we'll trace the loss of effectiveness of aging to the downregulation of 4 particular chemicals. We'll talk about what can be done currently, and obviously the research to extend our control over those 4 chemicals will be very important. Before going to that map, though, let me briefly run through what we know about aging currently. There are 4 basic processes that seem to be involved in aging. The first is, in the natural world, it's very rare for us to have enough calories. You talk about mice, they're restricted to 40% of the calories they would eat on their own, and they live longer. And the question, why do they live so much longer when they're only consuming 40% of the calories that they want? And the answer seems to be because that's the number of calories that they would get in the wild. The wild is a very, there isn't a lot of nutrition available. And our cells are designed to handle that, and in particular, so that metabolism does not come to a stop. What happens is when we do not have nutrients from the outside, our cells break down components of themselves, and particularly the damaged components, and reuse the molecules to reassemble new components. So there's this continuous process of self-renewal that's going on inside the cell. Officially, it's called autophagy. And in those rare moments in the natural world when there are enough calories present from the outside, autophagy, self-renewal, is set aside so that the cell can focus its energies on making maximum use of these external nutrients. And so in the natural world, cellular self-renewal is the norm. Nowadays, of course, cellular self-renewal is the exception because we have enough calories almost all the time. So one source of aging is this unnatural world that we live in now, where sufficient calories over nutrition are the norm. The second process we think is oxidative stress. These are byproducts of metabolic reactions and also of environmental pollutants, such as cigarette smoke. And we think that that gradually degrades the molecules in the cell and also creates an internal environment in the cell that disrupts chemical reactions. Oxidative stress was very promising back in previous decades. It led to the interest in antioxidants for preventing aging. It's been really tricky to make progress with oxidative stress. The fairest thing to say about it is, except for exogenous oxidative stress like smoking or being in a nuclear reactor, the oxidative stress that happens in our body to some extent is meant to be there, and it's really hard to parse out how to fine-tune that, and even if you can fine-tune it, to prevent aging. Nonetheless, we do believe oxidative stress plays some kind of a role. We just haven't really pinned down what that role is. The third is accumulated DNA damage. You can't run a cell if the basic blueprint is being degraded over time. And the fourth is loss not of genetic information, but of epigenetic information. The best way to, this is an oversimplification, but way of describing is this. So DNA is damaged, and it has to be repaired. And the cell has ways of doing it. The problem is the machinery that repairs the DNA cannot access the DNA to repair it if the DNA is being used, if the gene is being transcribed. So the first step in DNA repair is to take methyl groups, that's just a carbon with 3 hydrogens, and put it on the promoter region of the gene, and that prevents the gene from being transcribed. Now the gene is repaired, and we remove the methyl groups. However, not all of the methyl groups are removed. A few are left in place, and that's a signal to the cell, this gene was damaged and repaired, it may not be as good as new, be careful about using it. The problem is nature, and this is good, right? It protects against cancer when we're young. The problem is nature never expected us to live past the age of 30. And so this mechanism has no off switch. And so genes that are in regions of the DNA that are particularly prone to damage acquire more and more methyl groups. They become hypermethylated over time and become progressively switched off. There's some genes that by the time you're 90 are simply not functioning. One of those genes may account for 70% of the variance in aging. Okay, that's what we know so far. Now we're looking for a map that explains how metabolism controls aging. Well, that map has to do with energy generation. There are 3 stages of energy generation. The first stage is glycolysis that breaks down glucose into pyruvate, but we're not going to find the map there. For one thing, glycolysis only processes carbohydrates. For another thing, it's only 7 steps. It's not very flexible. It doesn't alter itself to changes in behavior. The last step is the electron transport chain that actually generates most of the ATP. But we're not going to find our map there because that's only 5 steps, and it's even less flexible than glycolysis. But in between is the Krebs cycle, also called the tricarboxylic acid cycle, discovered by Hans Krebs in 1937. And this has everything we want. It's 8 chemical reactions, 8 chemicals connected by 8 chemical reactions. Right? The 9th chemical reaction is the same as the first chemical reaction. It's a cycle. And it can speed up and it can slow down. It can go forward and break down molecules to produce energy, or it can go backwards and consume energy to produce molecules. Every molecule that's synthesized in the body, with one exception, is either made directly by the Krebs cycle or it's made by something that the Krebs cycle makes, made out of something that the Krebs cycle makes. The Krebs cycle, parts of it can go forward and parts of it can go backward at the same time. Or we can skip over parts of the Krebs cycle, or we can short circuit the Krebs cycle and jump over it. So it's tremendously flexible. When you eat an apple instead of a candy bar and it has healthy physiological effects, a lot of that is because you have altered the functioning of the Krebs cycle, and the Krebs cycle is altering the functioning of everything else. Okay, so the next step is I need to show you the Krebs cycle because that's our map. But at this point in the talk, I have to give you a trigger warning. I'm about to show you the Krebs cycle. So the Krebs cycle is one of the reasons I became a psychologist. There are great reasons for being a psychologist. It's a wonderful profession. Obviously, I don't need to tell any of you that. But there also is a not so great reason. So when I was in 19 and I was in an organic chem lab and there was a diagram of the Krebs cycle on the wall, I could tell from looking at it that it could only be understood by a different species of human being that had brain cells that were just not in my genetic lineage. And I don't know if it was if I actually walked out of the room or if it was metaphorical, but basically I left the room and changed majors. Now, my classmates were not so easily intimidated. And they stayed in class. They went on to have fairly unsatisfying careers as physicians. I think they wish they'd become psychologists. It's funny how things work out. And they do not look back on the Krebs cycle, obviously, as something to fear. For them, the Krebs cycle is, and this is a quote, that hellish thing I had to learn for the MCATs. So I'm a little nervous to show you the next slide, because I do not want anyone to walk out of the room, change careers, or remember this as that hellish talk I had to listen to at Journal Club. But remember, for us, the Krebs cycle is simply a map, and it's an important map because some directions in that map lead to a long and healthy life, and other directions lead to quite the opposite, so it's a map that really is worth our while knowing. Okay, I'm going to take a deep breath in, I'm going to let it out slowly, and here's the Krebs cycle. So it's 8 chemicals, and they're connected by 8 chemical reactions. You don't see the input here, but the citrate is made out of the input, which is acetyl coenzyme A, and the Krebs cycle just processes that input through 8 steps. This is not showing the actual enzymes. The more traditional presentation is here. Hope you can see that okay. Over at 3 o'clock is alpha-ketoglutarate. That's going to be important for us. Over at 7 o'clock or so is succinate. That's going to be important. And up at 11 o'clock, there's oxaloacetate. That's also important. So what made the Krebs cycle so fear-producing that it could change careers or be remembered harshly decades later? And I think the reason is, when you look at these chemicals, they're all things you never heard of before. I mean, who ever heard of alpha-ketoglutarate? Who ever heard of oxaloacetate in the 11 o'clock position or succinate in the 7 o'clock position? So the problem with the Krebs cycle, it was just straight memorization, arbitrary memorization, because the Krebs cycle was just not connected to anything. And that's ironic, because the Krebs cycle is connected to everything. Of those 6 trillion chemical reactions in the cell, as far as we know, every single one of them is connected to the Krebs cycle, and usually by only a few steps. And not just the cells in our body, but the cells in every living organism on the planet, even the most primitive bacteria. And not just the organisms that are on the planet now, but as far as we know, every organism that has ever been alive on the planet, even those that became extinct 1.5 billion years ago. As far as we know, a functioning Krebs cycle is an absolute biochemical requirement for something to be alive. Why is the Krebs cycle so central to life? To answer that, we have to first ask and answer an even more basic question. What does it mean that it's a cycle? So it's 8 enzymes, 8 chemicals. We can send away to a chemical supply company, order all of them, add them to a test tube, add water, and mix it up. And because each enzyme has only one particular type of molecule as its input and one particular molecule as its output, the chemicals immediately organize themselves into a cycle. And they start processing the input, acetyl coenzyme A, up there at 12 o'clock. And they continue processing until all the acetyl coenzyme A is used up. So the Krebs cycle is a self-organizing, self-starting, self-continuing system, even though it's not alive. But now we can go further. For each of the chemicals, let's say we decide in turn, each of the chemicals, we add 100 times too much of that chemical, or 100 times too little. And we're varying the concentration over a range of 10,000 to 1. And we're doing this because we want to know when the Krebs cycle breaks down, where does it break? At what chemical does it break? And this is what we find. The Krebs cycle does not break. If you add 100 times too little of a chemical, then the enzyme that produces that chemical speeds up because there's less product of the reaction to slow the reaction. If you add 100 times too much, the enzyme that processes that chemical speeds up because it has more raw material to work with. And within a few seconds, everything is back to its proper proportions. What is the most important invention in the history of the world? Is it writing? Is it electricity? Is it computers? Is it the printing press? I would say it's this. 4 billion years ago, in a world unrecognizably different from our own, the Krebs cycle invented homeostasis. And that became the foundation for the next great invention, which was life. So now we can understand the approach we're taking to aging on a deeper level. To do something about aging, we want to understand it at its most fundamental biochemical level. To do that, we need to understand life at its most fundamental biochemical level. And to do that, we need to go back in time to the moment on Earth before life first appeared, because there we will find the chemistry that became the scaffolding on which life was built and which remains the core of life to this moment. And if the Krebs cycle seems far away, consider that 2 of the enzymes of the Krebs cycle are the main producers of carbon dioxide in the body. The reason we are exhaling carbon dioxide at this moment is because of the Krebs cycle. The same Krebs cycle that was present 4 billion years ago, making it not only the most important invention in the world, but also the most successful. This is another view of the Krebs cycle, and it shows just a handful of the inputs and outputs. Okay, now it's our map, but how do we make sense of this map? What does this have to do with aging? Well, another way of thinking about it that I think is useful is to think of it as a crime scene. Because in the time that I've been giving this talk so far, about 4,000 people in the world have died. Some of them from old age, and even though, God willing, we didn't know any of them personally, they were still fellow human beings. I think what happened to them is kind of a crime. And we know from watching shows on television that the criminal is always at the crime scene. They're at the periphery, hanging out, just blending into the crowd. And so we're going to survey the periphery of the Krebs cycle, looking for a molecule that's just blending into the crowd. And we see it right away. Take a look up there at 11 o'clock, you see oxaloacetate, and now go straight left, straight west. You see aspartate? It's easy to overlook aspartate. It's the most nondescript amino acid. There's really nothing to say about aspartate until 2015, when 2 groups of researchers discovered that it was actually crucial to the reproduction of cancer cells. And biochemists were shocked because nobody expected aspartate to amount to anything. See, here's the thing with aspartate. Let's say you're dieting and you're hoping to burn fat. Of course, you don't burn fat, not right away, because biochemically that's complicated. The first thing the body does is it burns protein, because protein burns really easily. Protein breaks down into amino acids, and amino acids really easily burn. In fact, do I have an amino? Yeah, here's an example of an amino acid. So notice the C's, the carbon. Carbon burns great. And notice the hydrogen atoms, the H's, that burns great. So it's really easy to burn these, small amino acids that come from protein. There's only one catch, and that is, notice off to the left-hand side is NH3, an amino group. And that's nitrogen, and nitrogen does not burn. And so when we're burning protein for energy, which we do whenever we fast, the first thing we have to do is we have to remove that amino group. We have to remove the nitrogen. And that's a problem because NH3, when it's not attached to anything, is ammonia. And ammonia is a toxin. And so the ammonia has to be detoxified. And what the body does is it transforms it into urea, which is then excreted with the urine. And in the process of producing urea, aspartate is drawn down, aspartate is consumed. So when we are fasting, when we are dieting, when we are taking in fewer calories, aspartate levels go down. When we have plenty of calories, aspartate levels are high. But think, wait a second. Cellular self-renewal is blocked when calories are high. Does that mean that aspartate is what is blocking cellular self-renewal? And the answer is yes. Aspartate accelerates aging when it builds up, when aspartate builds up, by slowing cellular self-renewal. So now we know the first thing we want to do. We want to not produce so much aspartate. So notice that aspartate is produced out of oxaloacetate, which is just to its right. In fact, if you take oxaloacetate and you add an amino group, an NH3, it becomes aspartate. They're very closely related. So what we need to do is we need to give oxaloacetate something better to do with its time than to become aspartate. And notice we have an option here. Notice instead of going straight left from oxaloacetate, we can go kind of northwest. We can go diagonally up and to the left. A pathway that I've labeled here PepsiK, and I'll explain what that means later. And notice that one branch of that pathway goes up towards glucose, because this is how we produce glucose. We produce it out of oxaloacetate. When do we produce glucose? When we're fasting, when we're following a low carbohydrate diet. And so when we're fasting, we're producing glucose out of oxaloacetate, so we're not producing aspartate. Or we could follow that same pathway, but stop short of glucose. We could produce glycerol. When do we produce glycerol? Well, the muscles produce glycerol during endurance exercise. For the first hour or so, we're using glycogen, but after that, we have to produce glycerol. So when we're engaged in endurance exercise, we're not producing aspartate, we're producing glycerol, and we slow the rate of aging. Or we could follow that PepsiK pathway, but instead of going up towards glycerol and glucose, we could go to the left to glycine and serine, 2 amino acids. When do we produce those amino acids? When we're rebuilding muscle tissue, after resistance exercise, after weightlifting. And so now by looking at the Krebs cycle, looking at this map, we can see at a glance why endurance exercise, weightlifting, and fasting would slow the rate of aging, because they prevent producing too much aspartate. Now notice this pathway is called PepsiK. As you might have guessed, that's an abbreviation. It's an abbreviation for an enzyme. The full name of the enzyme is phosphoenol pyruvate carboxykinase, which is probably another reason why people do not like the Krebs cycle. But we abbreviated PepsiK, everybody does. What's important for us is that it is the first and the rate-limiting enzyme into this alternative pathway. So it is the valve, it is the gate. And so if there's a lot of PepsiK, or if it's very activated, then this alternative pathway is activated, and that should slow the rate of aging. In fact, it should slow it dramatically. Remember before I mentioned that mouse at Case Western Reserve University that lived twice as long because a single enzyme, gene encoding a single enzyme, was upregulated? This is the enzyme, PepsiK. We look at this now, it doesn't belong in this strange but true section of the news. It's obvious why the mice lived so long. They were not producing aspartate because this alternative pathway had its valve wide open. And so our first clue is that we want to we want to maintain the functioning of PepsiK. Unfortunately, of all of the projects, this is the trickiest. The regulation of PepsiK is very complicated, and it appears that it is upregulated during the reproductive years and downregulated after the reproductive years are over. It looks an awful lot like the body is downregulating that enzyme, whether that's a byproduct of something else or on purpose, we're not sure. I can say a couple of things. The first one is you cannot downregulate an enzyme and use it at the same time. So fasting, endurance exercise, resistance exercise, caloric restriction, they remain relevant to slow down the rate at which Pepsi K is downregulated. Secondly, Pepsi K is governed by cyclic AMP signaling, and that is upregulated by things like blueberries, ginger, nuts, resveratrol, a mushroom called forskolin. Those probably also slow the rate of decline of Pepsi K. Clearly, we want more research into how Pepsi K is regulated. Okay, so that explains overnutrition. What about oxidative stress? Well, none of the enzymes in the Krebs cycle are fans of oxidative stress, but take a look at the 2 o'clock position. You notice in small letters in italics is the word aconitase. That's a particular enzyme, and aconitase is particularly vulnerable to oxidative stress. In fact, it's so vulnerable that in elderly animals, that enzyme is just not functioning. And that's a big problem because if the Krebs cycle is essential for life, and if everything is blocked at the 2 o'clock position, kind of should be game over. But this is what happens. Take a look down at the 3 o'clock position, alpha-ketoglutarate. And notice that just to the right of alpha-ketoglutarate is glutamate. And there's plenty of glutamate in the cell. And glutamate, if we remove the amino group, so it's no longer an amino acid, it becomes alpha-ketoglutarate. And so it feeds the Krebs cycle through this side door at 3 o'clock. And so if 2 o'clock is blocked, that's okay. We'll just get this side door input at 3 o'clock. And that allows the Krebs cycle to continue through 4 o'clock and 5 o'clock and so on, and 8 o'clock and 9 o'clock, and 10 o'clock, 11 o'clock, and 12 o'clock, and 1 o'clock. But then we're stuck because 2 o'clock is still blocked. The aconitase is still blocked. So this is what we're going to do. We're going to take the glutamate just like before, and we're going to remove the amino group just like before. So now glutamate becomes alpha-ketoglutarate. It enters the Krebs cycle through the side door at 3 o'clock. But now we're going to take that amino group, and instead of throwing it away, or actually urinating it away, because that's what happens with amino groups, we're going to take that amino group and we're going to stick it onto oxaloacetate at the 11 o'clock position. And this is what the cell does. And it solves the problem, right? Because we now have input at 3 o'clock and output at 11 o'clock. And so all of the reactions from 3 o'clock to 11 o'clock can proceed unimpaired. Now that's still only half the Krebs cycle. And you say, well, is that enough? And I'll tell you, it's better to be 25 years old and playing with a full deck. But half a Krebs cycle is enough. And it's enough to maintain life. But there's a problem, and that is this was an emergency workaround, and there are usually some downsides to emergency workarounds in the long run. And this is no exception, because when we took that amino group off of glutamate and attached it across the Krebs cycle to oxaloacetate, it's not just that the oxaloacetate exited the Krebs cycle. The oxaloacetate became aspartate. The cost of staying alive is we just speeded up the rate of aging. And so now that's the problem, right? You're 90 years old, you're going to the gym 3 days a week, you're using the treadmill, you're lifting weights, you're still at ideal weight, you're fasting 1 day a week, but you're not getting out of it what you used to get out of it. You're not producing aspartate, but your body is, because aconitase at 2 o'clock is blocked. So our next clue is we need to protect aconitase. It's very rare, for an antioxidant to do that. There is one antioxidant that appears to, it doesn't do it all that well, but it kind of does, and that's the R form of alpha-lipoic acid. So alpha-lipoic acid probably would not do that much, if you're, a couch potato and you're eating the wrong foods. But if you're following a good metabolic lifestyle, then alpha-lipoic acid would have something unique to bring to the table, and that is that it would protect aconitase. Now, the last two things in aging, remember that the DNA gets damaged and the DNA gets hypermethylated. What does that have to do with the Krebs cycle? Well, it turns out there are enzymes in the body that repair DNA damage and that remove methyl groups. But both of those enzymes they're linked to the conversion of alpha-ketoglutarate at 3 o'clock into succinate at 6 o'clock. So they're linked to that other reaction, and that's all well and good. But notice that succinate is processed by succinate dehydrogenase at 7 o'clock. And succinate dehydrogenase is another enzyme that gets downregulated. Another reason why metabolic interventions lose their effectiveness. Succinate dehydrogenase is downregulated by oxidative stress, by aspartate, and so by overnutrition, and also by hypoxia, if our blood vessels are not functioning that well. And when succinate dehydrogenase malfunctions, succinate builds up. Now remember, DNA repair and DNA demethylation require converting alpha-ketoglutarate into succinate. But you cannot do that. You cannot produce more succinate if there's already too much succinate in the cell, which is exactly what happens when succinate dehydrogenase malfunctions. So the third thing we need to do to preserve the effectiveness of metabolic interventions is to protect succinate dehydrogenase. And alpha-lipoic acid should help with that. Maintaining good blood vessel health should help with that. Avoiding overnutrition should help with that, but this is another area where more research is needed. Now notice that aconitase, lastly, aconitase was damaged by oxidative stress. Succinate dehydrogenase was suppressed by oxidative stress. And so it's a big problem that oxidative stress increases with age. So why does oxidative stress increase with age? Well, really, we know the question is, what does the Krebs cycle do that causes oxidative stress to increase with age? Because the Krebs cycle runs everything. So, okay, let's say you want to prevent oxidative stress. So you take vitamin E. That's great, it's an antioxidant, but within an hour, it's all oxidized. It's not doing any good, so it has to be regenerated. How is it regenerated? It's returned to its antioxidant form by vitamin C. Well, that's all well and good, but now the vitamin C is oxidized. So how do we protect the vitamin C? Well, we use an antioxidant that the body produces itself, which is glutathione. Well, that's all well and good, but now the glutathione is all oxidized. So you see what's happening? None of these are actually antioxidants. They are links in a chain. They're messengers. They're a way of communicating antioxidant activity, but they don't cause the antioxidant activity. The glutathione is regenerated by an even more primitive enzyme called a molecule called NADPH. Now, the NADPH is produced by, let me go back here. Notice the input to the Krebs cycle, the front door to the Krebs cycle is at 12 o'clock. It's pyruvate. Where does the pyruvate come from? Some of it comes from glycolysis, the breakdown of glucose. But some of it comes from malic acid or malate that's in the cytoplasm. And when the Krebs cycle is going full bore, which in the natural world it always is, then it needs all the pyruvate it can get its hands on, and so a lot of malate is being transformed into pyruvate. And a side effect of transforming malate into pyruvate is to produce NADPH, which is the cell's fundamental antioxidant. All other antioxidants flow from that. And so when the Krebs cycle slows down and does not need so much pyruvate, oxidative stress rises because we're not producing as much NADPH. So we want to keep the Krebs cycle from slowing down. Now, part of it is to place demands on the Krebs cycle. That's why exercise is so important. That's why caloric restriction is so important. But that won't do us any good if the Krebs cycle cannot speed up, if the enzymes are damaged. And that also happens as we get older. But we can circumvent that to a degree by supplying increased amounts of coenzymes for these enzymes. And the coenzymes, the most important one is probably vitamin B5, pantothenic acid. But the others are B1, thiamine, B2, riboflavin, B3, niacin, and alpha-lipoic acid. I would not take megadoses of any of them because when you take pharmacological doses, the body reacts paradoxically. It can actually cause the equivalent of a deficit. It gets very unpredictable. I would not take much higher than RDA levels. I wouldn't take higher than RDA levels in the context of cancer because you might be helping the wrong cells to proliferate. But RDA levels make a lot of sense especially as one gets older, because at the very least, those allow the Krebs cycle to rise to the occasion and speed up. And when the Krebs cycle speeds up, that maintains our antioxidant defense. And so this is our map. Obviously, a lot more research is needed to understand how metabolism controls aging and what we can do about it, but there are some practical tools that we can see now. I'm certainly hoping that this helps the field move forward, and I hope that it helps all of us, all of you, move forward into a healthier lifestyle. I'll stop there.